Okay, so what I want to talk about is C3 versus C4 metabolism. We'll start with C3. C3 is basically what we looked at in the photosynthesis lab, and we're going to specifically look at the Calvin cycle. If you recall, the Calvin cycle begins with CO2. CO2 then comes in from the air, goes through a cycle, okay, and at the end of that cycle, it spits out sugar. Then the sugar is used in respiration for the plant and gives the plant energy. An enzyme or group of enzymes that's important in catalyzing this is called rubisco. And rubisco is the most abundant group of enzymes on the planet. It's also arguably the most important group of enzymes on the planet. The reason it's the most important group of enzymes on the planet is because it basically catalyzes this reaction, taking CO2 out of the air, utilizing ATP and NADPH. Utilizes that energy from light collection then to build sugars. Okay? Without sugars, then we don't have any energy for life itself, and heterotrophic organisms and so on and so forth can't survive. So rubisco is conceivably the most important enzyme on Earth. However, rubisco isn't a very well-designed enzyme. Its main problem is that in high CO2 it works great, but in low CO2 it does not work as well. Okay? Especially when out here we have the presence of O2 and CO2. In low CO2, rubisco has a tendency to take up O2 instead of CO2. When it takes up O2, it still runs a cycle, it still uses up ATP and NADPH. However, no sugar is produced at the end. Okay, so it's basically wasting energy when it takes up O2 instead of CO2. So it's not a really efficient enzyme in low CO2 concentrations. Okay, and this is a C3 pathway. In a C3 pathway, our O2 and our CO2 are both together here, where rubisco then can take up the O2 and the CO2. Okay, now we'll talk about the C4 pathway. Okay, so now let's talk about our C4 pathway. So here's our Calvin cycle from our C3 pathway. We know that when CO2 is in the presence of oxygen and we have low CO2 and oxygen together, rubisco does not work as efficiently. So what the C4 pathway has done is that instead of having rubisco work in the presence of oxygen and CO2, C4 pathway has a different enzyme. So what a C4 pathway does is it takes CO2 in from the air. From that CO2 in the air, it comes in and then it runs another cycle and then basically converts the CO2 from the air to malate and several compounds, and then it injects the CO2 directly into the Calvin cycle where we don't have oxygen around. The key enzyme then in the C4 is called pepcarboxylase. And so in a C4 plant then, where we have O2 and CO2 mixing around together, pepcarboxylase is the key enzyme then that fixes the CO2. And then the CO2 goes down, we change the compounds and injects the CO2 directly into rubisco, where rubisco doesn't have the problem with low CO2. Okay? So now for methods of this lab, what we do is we take a chamber, a respiration chamber. We place a plant material inside that respiration chamber, right? Then we shine light on the plant material. We close this off, and this is an enclosed system. So as photosynthesis occurs, then CO2 is going to go down inside this chamber because photosynthesis takes up CO2 from the air. Okay? As CO2 goes down in the chamber then, right? If we're looking at a C3 plant that is only doing rubisco, as CO2 goes down in the chamber, rubisco is not as efficient, therefore photosynthesis is going to decrease. And so what we're looking at then is in a C3 plant is the efficiency of rubisco in higher CO2 and lower CO2. For a C4 plant, then we'll be looking at the efficiency of pepcarboxylase in high CO2 and low CO2 for this experiment. Okay, now in lab, what we do is that you're going to be looking at a graph. That graph then has CO2 in parts per million on the y-axis, minutes on the x-axis. Okay? Photosynthesis then begins with higher CO2, and photosynthesis takes CO2 out of the chamber. Right? So we have our chambers here, right? CO2 is going to be decreasing in our chamber at the beginning of photosynthesis. Now for a C3 plant, right, we have a situation where as CO2 decreases, photosynthesis slows down. So when photosynthesis slows down, we're going to get a little bit of curve there, and then eventually it's going to go straight. Now the cool thing about this situation is that when photosynthesis slows down due to photorespiration in a C3 plant, we have respiration, cellular respiration occurring in the background all the time. Remember how we subtracted respiration from photosynthesis and respiration when we calculated photosynthesis. So in the background, this respiration occurs as photosynthesis slows down due to lower CO2. Respiration then is going to cause this to go up. When it goes up a little bit, photosynthesis speeds up because of higher CO2, then it goes down. Then respiration is faster, it goes up, then CO2 is faster, it goes down, and so on and so forth until it just keeps a straight line, and that straight line would go on forever. Okay? Now what we can do then is we can actually go across here and we can measure this value of CO2 that basically represents the point where photosynthesis equals respiration. Right? Now this is for a C3 plant. I want you to think about a C4 plant. What would happen to a C4 plant if we did a C4 plant? Now for a C4 plant, right, we would start with photosynthesis here. Photosynthesis would go down, but instead of decreasing at the same rate, a C4 plant, we're looking at PEP carboxylase, right? C3 plant, we're looking at rubisco. Rubisco has a problem with photorespiration. C4 plant, we're looking at PEP carboxylase. And PEP carboxylase then, if it does better in low CO2, our photosynthesis should stay the same and should actually keep going off at the same rate until CO2 itself becomes the limiting factor. And then we would have the same situation occur with respiration in the background. Thus, in a C4 plant, we would expect this leveling off to occur at a lower CO2 level than in a C3 plant. And that's what this experiment is going to be about.